idea is that at the end we will have vector which will contain our spike gene cloned inside this vector. Now what is going to happen, this ligation mix, because we are not sure this is cloned or not, so this ligation mix, this is our aim, this is our objective. So this ligation mix, vector plus insert in the presence of DNA ligase, we go to transform competent cells, bacteria. You remember our module in the genetic engineering. So in the bacteria are this potential vector after transformation of competent cells, because it has its own origin of replication. It will amplify, it will make many copies. And after transformation, we spread this on LB agar plates which contain kanamycin. You remember why we need kanamycin: to select only those plasmids, only those bacteria which contain our vector. So after plating and leaving them overnight, next morning when we come, we see single colonies. And these single colonies are the transformants. They are, so this step is called transformation. When you add ligation mix, so vector plus insert in the presence of ligase, when you add into bacteria, and in these competent cells, we simply heat shock. We give them heat shock at 42 degrees Celsius for just 90 seconds, and these DNA products, bacteria takes them up. After 90 seconds, we spread them on these plates. In the morning when we come, these colonies are called these are transformants, because these transformants, they contain our vector or vector plus insert. So now what we do, we take this, we pick up these individual colonies, and I refer you back to our earlier lecture or intro lecture on genetic engineering or recombinant DNA technology. So what we do, we add these colonies individually, random colonies you pick, and as I said earlier, I used to pick normally 6 colonies. And this media in these tubes, you have the same media, LB Loria broth, LB plus kanamycin. It's a liquid culture now, it's no more the solid plate. And in these cultures, you will see the bacteria growing after overnight incubation, overnight 37 degrees Celsius. So you incubate these bacteria in these LB containing tubes at 37 degrees Celsius overnight, and next morning when you come, the appearance of these tubes will be turbid, because the LB media is very clear. It's a very crystal clear, but in the morning, due to growth of bacteria, the media become turbid, and that's an indication that you have millions of bacteria growing in each one. Now, why we are doing this? Because we want to confirm that our ligation, the vector plus our spike gene insert, they are ligated in this form, and these bacteria, which are the transformants, they actually have spike cloned within the vector. So in this particular case, how we confirm this, we are going to, next day, after incubation at 37 degrees Celsius, we are going to isolate plasmid, and this plasmid is going to tell us after restriction digestion that cloning of spike is successful or no. So the next step is, of course, isolation of the plasmid from these cultures, and isolation of plasmid from these. Normally you have here maximum, we have 5 ml of LB in each tube. It's a small culture, okay? And isolation of plasmid from these cultures is called mini prep, because you are specifically isolating plasmid DNA. And once you have the plasmid DNA, then you perform the restriction digestion and confirm on agarose gel whether you could successfully clone the spike or no. Once you successfully clone spike here, then you subclone it in bacterial expression vector. So I stop this one here, and stay tuned. In the next video, we are going to start right from here, and we are going to do plasmid isolation from these mini prep cultures, these tubes, to confirm, because it is not yet confirmed, because these colonies, they are kanamycin resistant. They are kanamycin resistant, and kanamycin is on the vector. So these colonies, they may have just a self-circularized plasmid without the insert. So isolating plasmid from these cultures and digesting with our EQR1 XPA1, which are in our PCR primers, is very important. Otherwise, if you don't confirm, all these 6 cultures, they may have, or 5 of them may have just the vector minus insert. So next lecture is very important, and that will explain you the whole concept of DNA isolation, plasmid isolation from bacteria to confirm our plasmid. Okay, so stay tuned.